everybody, welcome to attend this talk. My name is Elsa Quesada and it's my real pleasure to present my project that I have been working in a JARREDI laboratory in Mexico, particularly in UNAM in León. Today my talk is titled ATG2 ATG18 complex during rhizobia and mycorrhizal infection process in legumes. First, I will talk about autophagy because here we find the ATG2 and ATG18 complex. Autophagy is a degradation process essential to maintain the cellular homeostasis. During the autophagy process, cytosolic components fall into a double membrane vesicle that fuse with lysosome or vacuoles where they are degraded and recycled. Autophagy is regulated by TOR kinase. Inhibition of TOR results in activation of constitutive autophagy. And autophagy is initiated when ATG13 is not hyperphosphorylated and results in ATG association. One of these complexes are ATG9, ATG2, and ATG18 and is involved in delivering the lipids for the expanding of a gopher. In the right picture, I show the autophagosome formation. Isolation membrane expand to form autophagosome by receiving lipids from endoplasmatic reticulum. This process is the essential for autophagosome form. Autophagy is related to a wide variety of physiological and pathophysiological roles as pollen development, cell germination, leaf senescence, root meristem, nitrogen and carbon starvation, anabiotic stress, and pathogen resistance. Little is known about autophagy in rhizobial and mycorrhizal symbiosis. And here I am showing some pictures of the main legume in our laboratory. It is the Phaseolus vulgaris and the different staging nodules from infection grid and mycorrhiza forming the arbusculum. To get started, we identified autophagy genes in 3 legumes. From 39 genes in Arabidopsis, we obtained 32 genes in Phaseolus vulgaris, 35 genes in Medicago truculata, and 62 genes in Glycemax. And here I show some of the complex ATG2, ATG18, and ATG9. We have 8 genes for ATG9, 4 genes for ATG2, and 30 for ATG18. To obtain the genes, we use BLAST and software to obtain orthologies with legume-based data, such as ensemble plant, cake, panther, and eggnog. These genes are tracked in the circus diagram where we find the Arabidopsis, Phaseolus vulgaris, Medicago truculata, and Glycemax chromosome with lines. We show the synteny relation and among genes family and that we obtain from Vista Synteny Viewer. Outside the cycle, we show the chromosome localization of the genes of the complex ATG2 and ATG18 and of course the ATG9. Then, as well as we obtained the gene protein sequence, also we obtained the promoter sequence. We consider 2000 base pair and with it we found the transcription factor in ATG2, ATG9, and ATG18 in plant care software. We found the MYB is the most abundant transcription factor in general, followed by MYC and BOX4. MYB is beta-proton gene involved in abscisic response. MYC is C-proton gene related with Hasmonate signaling, and BOX4 is involved in light responsiveness. In this graph, BOX4 bar, we can see the ATG2, ATG9, and ATG18 are abundant together, mainly ATG2 compared with another transcription factor bar. The results show the complex are involved in abiotic stress. Other study is expression profiling for ATG2, ATG18 complex with ATG9. First, the heat map contains part of the plant, and we appreciate the most abundant are ATG18A and ATG18G2. In the other heat map with our data, we compare mycorrhizae with nodulated roots. So the results are ATG2, ATG9, and ATG18G2 are upregulated in mycorrhiza. Meanwhile, for ATG18s are more expressed in nodulated roots. The third heat map is to show the expression of ATGs in roots and nodules in efficient and inefficient nodulation. Both some ATGs have high expression efficient nodulation, and we want to understand this situation. So I will show some studies in different levels in these three components of the complex to form the autophagus membrane. Well, the next slide. As I said earlier, ATG2 reach a highly cured liposome, thereby facilitating efficient phospholipid transfer. In the figure is an example in Saccharomyces. ATG2 in the blue box show how to the phospholipid move from the endoplasmatic reticle to autophagus. For ATG2, only I show these models, and we can see the small figure are using Arabidopsis data, and ATG2 is related with ATG9 only with ATG18A. However, when we generate the same diagram in Fasciolus vulgaris, the model change and appear ATG18B and ATG18C. This means we need to explore more about the relation into ATG2 at least 6 elements of ATG18 in Fasciolus vulgaris. Next, we found the ATG18 genes and proteins in 13 legumes, 3 monocots, and 2 model plants. The total genes are 115 and proteins are 270. We explored the ATG18G2 that appear in the expression profile in efficient and inefficient nodules in Fasciolus vulgaris, and it is the same in Medicago truculata and Lysine max. Here I show the 3D structure of ATG18G2 with the common structure and WD protein containing symmetrical plates. In the other level, we have the promoter construction of ATG18 in Fasciolus vulgaris that will help us to understand the autophagus membrane dynamics. These results show the expression in meristematic zone and vascular tissue in roots with 6 days. Meanwhile, the expression of ATG9 nodulation appear at the same place in vascular tissue. In early stage in the expression appear between the nodule and root, and we can see the vascular expression in efficient nodule, and finally the expression of ATG9 again in the vascular expression but in senescent nodule. Finally, in this talk, I show you some studies in 3 different families which are part of the complex to understand the role of autophagy. In conclusion, our autophagy genes identification include 8 genes of ATG9, 4 genes in ATG2, 13 genes for ATG18 in Fasciolus vulgaris, Medicago trunculata, and Lysine max. Promoter studies suggest ATG2, ATG18, and ATG9 are involved in abiotic stress. The quantitative expression profile by RNA sequencing identify ATG2, ATG9, and ATG18G2 are upregulated in mycorrhizal root, and for ATG18B, B, F, and G are regulated in nodulation root. In ATG2 in silico interaction show different relation with ATG18 proteins. ATG and 18 appear at least 6 genes in legumes and a lot of 17 different ATG18s. ATG9 expression appear in meristem, vascular tissue in roots and nodules of Fasciolus vulgaris. The majority of ATG18 is a key to understand the different response, but ATG18 requires ATG2 and ATG9 to form the autophagosome to maintain the homeostasis. Well, thank you to my advisor to support me, to all the laboratory members, and for the events. Thank you and share for Mexico.